mechanisms that maintain this unique structure and how alterations in the ciliary gene can lead to photoreceptor pathology. Moreover, her investigations into the process of outer segment disc renewal offers critical insights into both normal physiology and disease states. In addition to her groundbreaking research, Dr. Pearring is a passionate advocate for academic mentorship and diversity in STEM. At the University of Michigan, she plays an active role in graduate admissions, serves on rotation advisory panels and dissertation committees, and has personally mentored over 20 trainees. Her impressive record includes over 30 peer-reviewed publications and the leadership of research supported by 2 active R01 grants, along with several prestigious awards such as Individual Investigator Research Award from the Foundation for Fighting Blindness and the Faculty Service Award from the CMB Graduate Program. It's now my great pleasure to invite Dr. Pearring to deliver her talk titled Disrupting the Ciliary Gradient of ARL3 GTP Activity Results in Photoreceptor Nuclear Migration Defect. Please join me in warmly welcoming Dr. Pearring.
Great. Thank you very much. That was a great introduction. I appreciate that very much. And I'm very happy to be here with everyone today. I'm looking forward to telling you sort of a little bit of a different segue from the work that I've been doing in the past, where we actually identified this very unique phenotype where we have a nuclear migration defect in photoreceptors. So let's get started. I can do that. Here we go. So I know that this audience we don't have to actually do much introduction to the eye, but I just want to sort of start by really drawing our attention to the retina, which is the neural piece of tissue lining the back of the eye. And I really want to focus our attention today on the beautiful laminated structure that is the retina. And this is common amongst all of the CNS, where you have a very nice layered structure, which is important for signal transduction, right? We know that the light-sensitive photoreceptors occupy this outer nuclear layer here, and that they then transmit the signal to downstream inner retina neurons sitting in the inner nuclear layer here that help to sort of modulate the signal. And then it's summated by the ganglion cells, which form another layer here in the inner retina that then send that process signal out to the brain. And it's important to maintain all of these different laminated structures and ensure that each different cell type is occupied in the correct laminal position. Interestingly, this sort of layered structure can actually also be seen if you just look at a photoreceptor. And this is really what I've been spending most of my career looking at, is the photoreceptor itself. And you can see beautifully that the photoreceptor is nicely polarized, and actually different cellular compartments actually reside in these very distinct layers that you can shown here too. The most apical side of the photoreceptor is the outer segment. This is a membrane-rich modified primary cilium, and this is what houses all of the visual signaling components for light capture and signal propagation. Then you have that connected to the inner segment, which is sort of the housekeeping part of the cell. This is where you have mitochondria, ER, and this is where you're going to have energy production and lipid and protein synthesis. Then you have a big stack of nuclei. This is all coming from sort of mouse retinas here. We have about 10 of them stacked up on top of each other. And of course, the nuclei is where all the genetic material is being housed. And then at the very basal side of the photoreceptor, you have the synaptic terminal, which is going to be what's actually responsible for sending the signal to the downstream interneurons in the next layer, in the internuclear layer. And when I was a postdoc with Vadim Marshofsky at Duke, I spent a lot of energy and time looking at how transmembrane proteins and signaling molecules that were initially synthesized here in the inner segment, how they were being properly delivered into the ciliary outer segment here for the outer segment, things like rhodopsin and peripherin. These are very critical components of phototransduction and are important for sort of structural organization of this compartment. And so when I started my own lab in 2018, I thought that a natural segue would be to start thinking about lipidated protein transport into the ciliary outer segment. And so what we know is that lipidated proteins are actually being first lipidated in the inner segment on intracellular membranes like the ER. That's where the prenylation proteins are actually residing and attaching lipidated moieties onto these lipidated proteins. And lipidated proteins can actually dissociate from the membrane sort of in a stochastic way, and they can be picked up by these GDI-like chaperone proteins that I'm showing here with this like little blue chaperone here. And basically, the chaperone proteins do a really nice job of sequestering the lipid moiety on the inside cavity of that inside cavity of the chaperone, so that as a complex, these two proteins can then solubly sort of diffuse through the cytoplasm, right? And some beautiful work from Wolfgang Baer and others have shown that proper enrichment of these lipidated proteins into the ciliary membrane of the outer segment is really regulated by this small GTPase called RL3. And so the small GTPase RL3 can actually, when it's active in its GTP state, will bind to different effectors. And some of the effectors it binds are these GDI chaperones. And when they bind to the chaperone, then the lipid moiety and the lipidated proteins will be released from these chaperones, and then they'll be attached to the nearby membranes. And so what's unique about RL3 is that it is only being activated in the ciliary outer segment. And that's because that's where its guanine exchange factor, its GEF, which actually helps to activate or perform nucleotide exchange for RL3 resides. So you have a very sort of unique situation where you're only activating the small GTPase RL3 within the ciliary compartment or the outer segment, because that's where RL13 is sitting. And outside of that compartment, RL3 is actually being inactivated by its gap protein, which is RP2. And so RP2 sits in the intersegment plasma membrane. And so you have this sort of nice system where you have activity of RL3 localized to the outer segment and inactive outside of the outer segment. And it was shown beautifully that this is really helping to regulate lipidated protein transport, which is really critical for phototransduction. There's a number of components of lipidated proteins, such as transducin and the GRK1 and things like that, that must be enriched into that compartment for function. So when I first started the lab, there was a number of patient mutations that were being shown to come out in RL3 that caused retinal degeneration. And so here I'm just showing you a crystal structure of RL3, and I've put down some of the different patient mutations sort of on the crystal structure. And all of the green mutations were actually autosomal recessive mutations. So that means you needed to have two bad copies to cause disease. But there was two mutations that were being described as autosomal dominant, where they only had one bad copy. And I'm showing these ones in blue, this Y90C and D67V. So you only need one bad copy in order to cause disease. And just as a side note, there's been a third autosomal dominant mutation that was published just last week, and I need to update my cartoon, but I'm not there yet. And this was done by Alfred de Beer and Michael Cheatham. So it's pretty exciting that more of these are being identified. So we sort of started to ask the question, right? We have sort of two classes of mutations. And so like you can imagine that because of this, it would really suggest that recessive mutations may actually have a different pathology from the dominant mutations, right? That recessive ones would be maybe causing a loss of function, and dominant ones may be having a dominant negative effect. So we wanted to be able to try to test this in the photoreceptor and see if we can't figure out what might be different about these two different classes of mutations. And so a postdoc in my lab, Amanda Travis, took on this project and really wanted to ask the question about, are these dominant mutations having a distinct pathology? And just some evidence that this may be the case came from the literature initially. And that really came from the fact that from mouse studies where people had done some really beautiful work where they had knocked out RL3 and rod photoreceptors. And we knew that if you knock out RL3 from rod photoreceptors, specifically, you lose the rod photoreceptors. You have a pretty nice drastic loss of rod photoreceptors over time. And in fact, you can also get the same phenotype by overexpressing a constitutively active RL3 mutation as well. And this was done here in the bottom, also causing retinal degeneration, right? So this also suggested that you kind of had two different ways to cause disease, either completely losing RL3 function or by sort of affecting the way that RL3 is behaving in the cells. What was interesting, though, about this constitutively active transgenic mouse was it also had a unique phenotype where a number of the photoreceptors were actually mislocalized into the internuclear layer. So they're just driving this transgene of constitutively active RL3. It's called Q71L. And they can see that now you have some of these photoreceptors not making it into the proper layer. So that was really quite interesting. And it also kind of made us start to think a little bit more about RL3's function outside of like sort of mature photoreceptors, right? So most of the field had really looked at RL3 within a mature context when you have a fully formed outer segment and trying to get these phototransduction lipidated proteins into that ciliary compartment. But we knew that RL3 is also important for enriching lipidated proteins into any cilia, even just in a cell culture model system. And so we started to think a little bit about maybe RL3 had playing a role during development, because that's actually when we know these nuclear migration, the nuclear migration is occurring, right? So in mice, it's quite nice that postnatally, we still have a lot of retinal development occurring. A number of rods are still being produced at P0, and all of these new cells are being born at the apical neuroblast layer here. And then they actually have to migrate into their correct layer. So you can see here this nice image with a P3 staining for different cell populations, that you have sort of a little bit of a mixture here. You haven't quite formed your layers yet. But then when you go to a mature retina here, all of your photoreceptors are now nicely positioned in the outer nuclear layer like this. And so we started to think a little bit about RL3's function before the outer segment is actually formed, and how it might be helping to establish some of these, how to maybe establish some of these layers. A very nice paper also came out while we were doing this work, looking at some of these early movements of the sort of baby rod photoreceptors right before they have their outer segments. This is early postnatal time points, and they were looking at the apical and basal trajectories of the nucleus and monitoring this with live cell imaging. And this was a very nice paper because it kind of helped us to think a little bit about how important these nuclear movements are for getting photoreceptors into the correct layer. What they also showed in this paper beautifully was that if they knock down dynein components, so these are molecular motors that usually hook onto microtubules and help to walk organelles around the cell, if you actually knocked out dynein, you had a similar phenotype to what they showed with this constitutively active RL3, where now a number of the rod photoreceptors are sort of sitting basally and even displaced into the internuclear layer. So this was quite interesting to us, that we're sort of seeing this sort of phenotype occurring in different ways. And so we really wanted to ask, do the dominant human pathogenic mutations of RL3, this Y90C, this D67V, do they disrupt normal rod migration? And in our lab, we use an in vivo electroporation technique to do this. So what we do with in vivo electroporation is we're able to sort of transfect baby rod photoreceptors at P0 right as they're being born. We can use rod promoters to drive expression into those cells. And then we can actually look at that population of cells and ask how they're behaving. We actually put the pups back and let them develop the retinas just normally. And then we usually are harvesting all the tissue at about P21 when all the layers are nicely established. And what we can see as cell biologists, I love this technique because we can see individual cells labeled. Here, we're just labeling full-length RL3 with a 3X flag tag, and we're just labeling for flag, and you can see RL3 is nicely distributed throughout the entire cell here, and we can see individually transfected cells like this, which is quite beautiful. So my postdoc Amanda did a very nice thing, and she wrote this beautiful code so she could actually quantify the position of every single nuclei that we had labeled. So all the cells that have been transfected gets a little mark, and then she can look across all the different eyes that we've electroporated and be able to get a sense of where the different nuclei are sitting within the outer nuclear layer. So here you're going to see below a lot of these, a lot of the data moving forward, you're going to see that we have this nice sort of dot plot showing you the position of all the nuclei from each electroporated construct. And so this is just showing you full length RL3. This is wild type conditions, and we have all of the nuclei where they belong within the outer nuclear layer. Here, 0 is sort of representing the border here at the basal side, and 1 is the apical border here. And indeed, when we expressed the dominant mutations, D67V or Y90C, we saw this very interesting pattern where now our dot plots look a little bit like Christmas trees, and we see that we have basal distribution of rod photoreceptors, right? All of the nuclei are sort of sitting low within the outer nuclear layer, and some of them have actually jumped the border and are sitting in the internuclear layer, right? So this was quite interesting to us. We got this with both of these different human mutations. And we're very excited, and we wanted to show that this could also be done similarly, that we could get the similar results that we saw with the transgenic Q71L mouse, which we could do here. Again, we just expressed in our electroporation, this Q71L is a very typically used constitutively active. That means that the RL3 is sort of locked in a GTP state. And we have the same sort of Christmas tree presentation. But if we tried a different RL3 mutation, this is T31N. What that means is this mutation prevents activity. So you actually have a GDP bound RL3 that cannot be active. We no longer had this displacement. And so we really wanted to ask, are these human mutations really just causing RL3 to be constitutively active now? So is this just recapitulating the same mutation that we saw in the transgenic mouse? And so that was seen in the transgenic mouse. So Amanda decided to do some biochemistry to start to like tease this apart a bit. It's lovely to use the enzyme sometimes, right? As a biochemist. All right. So as a biochemist, what we do is you express these things in hex cells, right? And then you lyse open the cells and you ask whether or not something is locked in a GTP state by looking at whether or not it can actually bind to its effectors. So one of the effectors is PDE delta. This is one of these chaperone proteins that helps to actually escort lipidated cargoes to the outer segment. And we just use PDE delta on a GST column. And then we put our lysates over and ask the question, does it bind to the PDE delta? And you can see with a constitutively active RL3, you have very nice binding here in the elution, but you don't see any binding of our inactive mutant shown here. And so when we looked at our two human patient mutations, what we actually saw is we had very strong binding, similar to a constitutively active Q71L for the D67V, but we did not get binding with the Y90C mutation, which was pretty surprising to us. So is Y90C not constitutively active? I mean, this is what this assay is telling us. And so we wanted to ask, like, can Y90C even bind to GTP? Because our phenotype suggests that it has a lot of activity, right? That's what the cell is telling us. And so we decided that we would try to just see if we could push the system in a way to make Y90C bind to GTP. And so in these assays, what you oftentimes do is you have magnesium sitting in your buffer. And that's because magnesium is used as sort of a coordinating ion on the inside of the small GTPase and helps to hold the nucleotide in place. Basically, what a GEF is doing is just kicking the magnesium out of that inner pocket, allowing the nucleotide to be released. So you can kind of think of magnesium as sort of a little lock on the nucleotide. So if you add EDTA, you can actually chelate away all the magnesium in your lysate, and that allows now any nucleotide to bind into your small GTPase. And so if you just load a lot of GTP, you can ask the question, does my small GTPase, bind to GTP, right? I can just sort of force it in there by taking away the magnesium. And so when we did that, we could see very strong binding onto PDE delta of our 3X flag tagged RL3. And here we're actually blotting our Western blots with an RL3 antibody. So you can see endogenous RL3 here in the band below. And so we get very nice strong binding as long as we have enough GTP in the system, right, that we sort of added to the system with EDTA. But interestingly, when we were running the control with magnesium still there and just adding a lot of extra GTP, we saw that this Y90C could also still bind to its effector, right, suggesting that just in the presence of GTP, this Y90C was being loaded, right, could be loaded even though you had magnesium supposedly locking it so it shouldn't be loaded, right? So it was a little bit of a confusion because this is not typically what happens is, as you can see with the endogenous, you don't usually have GTP binding when you have the presence of magnesium. And what is actually interesting is when we started looking in the literature, this is a very common phenomenon for mutations called fast cycling mutations. And there was another just let the nucleotide fall in and fall out, and they could cycle quite quickly. And so we wanted to test that we were getting a similar kind of result with another fast cycling mutation, this D129N. And indeed, in our system, again, we see the same thing where as long as you have enough GTP around, that small RL3 is going to be active and bind to its effectors. So we then thought, okay, well, that's pretty cool. do fast cycling mutations cause this migration defect? And so we wanted to test this D129N in the photoreceptors. And indeed, when we electroporated this into photoreceptors, we saw that this had the same migration defect as we saw with Y90C shown here. So indeed, being a fast cycling RL3 mutant would be enough to cause this migration defect. But how else do we know that RL3 is really being like a fast cycling mutant inside the cells, right? All this is just really done in a test tube, and we're really forcing the issue by adding a whole bunch of extra GTP, right? And so we started to think a little bit more about what are the common features of a fast cycling small GTPase, right? They really have two important features for their function. One is the one I've been talking about, which is that they have this ability to exchange nucleotides even in the absence of the GEF, right? So usually the GEF is kicking out the magnesium, but these guys can actually become active at any point. Even with magnesium still there, they're becoming active, right? So are these Y90C doing this function in cells? And also the other thing is we know that because they have this propensity to lose the nucleotide so frequently, they also actually are in a state where they don't have nucleotide more often than other small GTPases. And they actually will bind to, inappropriately bind to the GEF itself, because the GEF is usually bound to it when it's sort of kicked the magnesium out and doesn't have any nucleotide. That's sort of when they're there. And indeed, this D129N mutation that I brought up was actually used to identify RL3 initially as the GEF for these RL3, RL13B as the GEF for RL3 initially. So you oftentimes use these fast cycling small GTPase mutations in order to sort of identify the GEF, because they're a great binding to the GEF. So we wanted to sort of test these two features. So the first one was, how are we going to actually look at the state of RL3 and its activity in the cell, right? And so we came up with a beautiful idea where we would use in vivo cross-linking, right? So we can actually express this into our hex cells, our different constructs of interest in the hex cells. We can use this permeable cross-linker to add, so we can actually cross-link RL3 to any of its effectors that it's bound to, right? And if RL3 is overly active, it's going to be more likely to bind to its effectors. And then we can use our flag tagged IP to pull out that RL3 and look for heavier weights, molecular weights, where it's actually bound to the effectors in that cell. So this was sort of the idea, and we wanted to test this initially just with our two sort of standards here, where our inactive mutant and our active Q71L, because it's really active. And I know it looks like a lot. Obviously, when you cross-link things, get a little bit dirty, but we did mass spec on this to confirm that these were the different bands that we had from these cells. And basically, we identified three components that this constitutively active RL3 can bind to in cells, which is RP2, UNC119, and this BART protein, which is actually an adapter with the GEF RL13B. And so these are sort of our standards as saying, okay, if you have activity in the cell, you're going to see these shifts in molecular weight on our Western blot under these cross-linking conditions. And that's a great way to sort of take a snapshot of activity within the actual cell. So let's take a look at what happens when we look at Y90C. So here again, we're looking at these same bands, the BART, the UNC, and the RP2. And we saw that with Y90C, we were not having very much binding to RP2. And that's actually what we kind of expected, because RP2 typically is a very transient thing. It binds and it hydrolyzes very fast. The fact that it's able to bind to Q71L is actually a product of Q71L being overly active and locked in a GTP state. It sort of binds and tethers itself to RP2, so RP2 can't be released. And so we didn't really expect to see RP2 in these lysates, and that was accurate. But we could still see elevated amounts of UNC119 and this BART protein in both the Y90C and then our control, the D129N fast cycling. And when we quantified this across many blots, we could see that the activity of Y90C was indeed elevated compared to a wild type RL3, which was not significantly different from our inactive T31N here. So this really suggested that there is more activity, RL3 activity inside the cell with the Y90C mutation. Okay, so we've got a little green light here. And so then the next question is, do we see more binding to RL13B, the GEF? So this is pretty straightforward. We can just do straight co-immunoprecipitations. We overexpressed RL13B with a GFP tag here. And then we pull down and look to see whether or not we had binding, right? So we co-expressed our RL3 constructs with this RL13B, and we can see quite nicely we get a very nice strong binding in our Y90C mutant. similarly, we could get binding with this other D129N. So that really does suggest that we do still that RL3, where was I headed? Okay, RL3 does bind to RL13B. But then the question is, okay, if it's sitting there bound to RL13B, does it actually prevent activation of the endogenous RL3 that might be sort of in the cell normally? And we thought we could probably use this cross-linking assay to get to this question, right? If we express our wild type flag tagged RL3 in cells, and we express our RL13B GFP tag, we can see that usually you have our activation of wild type here, binding to UNC119 and BART. But if we now also co-express a Y90C mutant into the system, we saw that these two bands disappeared, suggesting that UNC119, sorry, I keep looking at this, suggesting that Y90C is actually binding onto RL13B, right? And blocking its ability to activate the endogenous or wild type, which we're showing here with the flag tagged wild type RL3 here. So we're actually getting less activity of the wild type when you have Y90C in the cells. So then we thought, okay, well then maybe we can rescue the Y90C's phenotype by just giving it more RL13B, right? If we sort of provide more here, then maybe we can outcompete the Y90C binding and be able to restore a bit of this cycle of the wild type and get our nuclei to migrate properly. And that's exactly what we were able to find in our photoreceptors. If we co-express the Y90C mutant with RL13B now, which we know is expressed in the outer segments up here. I didn't show that, but that's where it's located. It's beautiful. And you can actually get this nuclear migration restored because you're now sort of tipping the balance so that wild type RL3 is able to recycle and be active in the correct location. And so with that, we think we found that Y90C shows both features of fast cycling small GTPase within the cell. And we sort of started having this sort of working model for different ways that were causing the pathology from these two different human mutations. One being this sort of fast cycling that's disrupting the balance. And here I'm just showing my photoreceptors as sort of my nascent baby photoreceptors. They don't have outer segments yet. So I just have a cilia here sitting at the top. And really, I'm using green to sort of highlight activity. So active RL3 typically sitting in the cilia, inactive sitting in the cell body. And then with the Y90C, we have sort of this bit of a conundrum where we're reducing the amount of active RL3 in the cilia by sequestering the RL13B with the Y90C mutant. But we're also having an elevated amount of Y90C being able to actually exchange and have GTP within the cell. So we kind of termed this as orange because we can imagine there's a little bit of a mix-up. There's a lot of different things happening here. But with our constitutively active, we imagine that everything becomes overly active. We know that this Q71L and the D67B both bind strongly to RP2 that blocks normal hydrolysis of active RL3 in wild type endogenous. And so you would have just sort of a very strong overly active RL3 situation. And so we really started to ask the question, okay, so is this migration defect really caused by all of this aberrant RL3 activity in the cell? Like is just having too much RL3 GTP sitting in your cell what's causing this migration phenotype? And we wanted to sort of ask the question of like, if we just took away all of RL3's activity, would we have a migration phenotype? Because the knockout mouse of RL3 didn't show this phenotype of a mismigrated rod photoreceptor nuclei at sort of the full body knockout level or full rod knockout level. And so we asked, like, is there a way that we could do this in our electroporation technique in our wild type mice, right? We have a lot of different mutants that we can go to. And so we started thinking a little bit about all the different mutations we have. And if we mixed the T31N, which we know is an inactivating, unable to bind GTP mutation of RL3, with the Y90C mutation, which is going to bind onto RL13B and prevent normal activation of any endogenous wild type RL3 in the cell, we sort of postulated this could potentially create a situation where we would actually block all of RL3's activity in the photoreceptor. And we were confirming some of this with our in vitro assays from hex cells showing that indeed this double mutant would still bind to RL13B, and indeed it did no longer had any activity for its effectors in this cell type. And so when we actually expressed this into our photoreceptors, we can see that indeed we prevented the nuclear migration phenotype. So it does appear that, blocking RL3 activity does not affect migration, right? So a complete knockout of RL3 is not going to have the same migratory effect that having overly active is. So there really was a very interesting idea of like, okay, these two dominant and recessive mutations are sort of really distinguishing themselves in a different way, and that having some sort of overly active component to RL3 is what's driving the phenotype. And we thought, okay, what else can we learn about this phenotype? And can we learn more about it by doing some rescue experiments? Because we're small GTPase people and we're thinking hard about this cycle here, and about what would happen if you have an increase in nuclear of RL3 activity in the cell, right? So if you have too much RL3 activity, what ends up happening, you can envision, is that now you're binding to your effectors where you shouldn't. It's sort of aberrant activity all over the cell. And now your chaperones, your effector chaperones can't pick up your lipidated cargo at the membrane properly. And then you can imagine you'd have a defect in lipidated transport into the cilia. So could we just restore this by giving the cell a whole heck of a lot more chaperone, right? Like sort of using it as almost like a sponge to soak up the overly active RL3. Since we know that, even if you don't have RL3 activity, it cells okay with that. Like they can still migrate. So let's just see if we can just soak up some of that overly active RL3 and sort of tip the balance back to picking up cargoes, right? And so we tried that by overexpressing both UNC119 and PDE delta. These are both RL3 chaperones for cargoes and RL3 effectors. And you can see that overexpression of these with Y90C beautifully rescued the nuclear migration phenotype. So now all the rod photoreceptors are sitting back in the outer nuclear layer, which was quite nice. Okay, so at this point, we sort of come up with this nice model where we had imagined that we're sort of using PDE delta to sequester any of the overly active RL3 in the cells. We're definitely probably doing that here at the cell body level. We could also potentially be doing it at the actual level of RL13B. It's hard to say within cellular context. And so that started to make us think a little bit about, how important is just having a gradient between these two compartments, knowing that you have activity really in one place and no activity in another place. Like, is it really the aberrant activity of RL3 outside of the cell? Or is it just having a change in activity between these two compartments that's helping to drive lipidated transport into the outer segment, or, well, the primary cilia at this point? And so we thought, could we actually restore the defect by just restoring this gradient? And so we decided to try a mutation that actually causes RL3 to not be able to bind to the GEF RL13B. So this is an R149H mutation, and this is shown to be between the interface between RL3 and RL13B. And now these two can't bind to each other. And so we can envision if we stop the binding of RL3 onto RL13B, then we would restore normal wild type's ability to be activated in that compartment. However, we would know that the overly active ability of RL3 Y90C is still sitting in here in this mutation because it should still be able to actually bind to a lot of GTP. And so we just confirmed here on our Westerns again that we have prevented binding of RL13B with this mutation shown here. And we also showed that we were able to still maintain the overly active nature of Y90C using our cross-linking assay shown here. And so this suggests that that we wanted to ask the question, of course, then if we just put the activity of RL3 back in the cilia, will we be able to restore the migration defect? And we did show, we were able to find that indeed that did happen. So even though we still had overly active RL3 sitting in the cell body, as long as we maintained a gradient and had enough activity back in the cilia, we were able to restore this nuclear migration phenotype, restore nuclear migration of the rod nuclei. So at this point, that was sort of the culmination at the end of the paper, right, as we wrote. We had a couple of things we really feel like we were able to find and sort of nail down, which is that dominant mutations in RL3 really cause a beautiful developmental phenotype, migration phenotype. And then, and that we have sort of two distinct ways of getting there. You can do it through constitutively active, or you can do it through fast cycling, and that you can actually overexpress chaperones in order to get this to be restored. So this was really, really great. And I can take questions now, or I can keep going because I have like sort of the next session. But yes, absolutely, questions.
Let me ask you, because I think I'm lost a little bit.
It's a lot of
it's embarrassing to ask, but anyway, I wanted to learn because it's a really beautiful work you're doing. Those chaperone proteins, like delta, right? It is involved in PDE or maybe kinase to transport.
Yep.
And that's understandable. But why nuclei will migrate in a different place?
Yeah.
This has nothing to do with the cargo.
Well, that's what we're trying to get a handle on. We think that there probably is a ciliary signal during these early developmental time points that may be directing nuclear migration. And so I think classically, we think a lot about PDE and UNC119 in their roles with phototransduction components, but they absolutely play critical roles in delivery of lipidated proteins to primary cilia as well. And we know that the postnatal rods at these time point have a primary cilia sitting there, right? Nobody knows whether it has signaling capacity. Nobody knows what might be sitting in there that to help direct these migrations. But I think that our data is really suggesting that there is some sort of cargo that RL3 is helping to deliver into the cilia in sort of these baby rod time point, right? This sort of developmental time point that is then telling the nuclei which direction to go. And we're really
that pathway to nuclei is completely unknown.
Completely unknown. Yeah. So the only work that really showed anything was that dynein is absolutely involved, and that would make a lot of sense. Dynein is a molecular motor that moves large cargoes, moves things towards the centrosome. You have the centrosome sitting at the top of the cell, so that would make a lot of sense that dynein movements would actually help sort of regulate the apical translocations as you sort of pulling on rods. But the rest of the signaling is completely unknown, I would say. Yes.
Okay, so your assay really directly not measuring the movement of these lipidated phototransduction proteins, but rather a surrogate assay of looking on nuclei movement.
Correct.
Okay.
Yeah. So at the moment, we're really.
I thought that I'm dreaming.
No. At the moment, we're really sort of poking at the small GTPase, right? We're poking at the cycle. We're poking at sort of what is downstream in some ways. I guess you could think of it as upstream too, as to what actually gets it into the cilia. And we're hoping, in the next section, I hope to get there to find the actual signal. But
so let me have one more question.
Yeah.
You mentioned that cilia, not only in photoreceptors but in other systems, the same small G protein is involved in the migration on. Where the specificity come from? I mean, you don't have that many adapter proteins that bind those.
Right.
And yet they show some specificity in biochemical assays.
Yes, absolutely. Yes. So there's a lot of different RL small GTPases, and RL is sort of short for ARF-like even. So there's even a larger class of ARF-like small GTPases. But RL3 seems to be the only one to actually be doing these cilia directed. And I think a lot of that is because RL13B is the specificity for RL3. Like that is how RL3 is being activated. And so that really lends it to, ensuring that its activity is in a particular location, which is the cilia. RL13B is like probably the most classic ciliary marker that people use in cell culture and everywhere just to like find a cilia, right? That's oftentimes what people will stain for because it's so striking and so specifically unique. So I think that is where the specificity is really coming from. And then in terms of the chaperones, they have specific cargoes that they like, whether you're mysterolated or prenylated or geranylated, you'll bind to different chaperones. But a lot of these chaperones can actually bind to cargoes that need to go to all different locations. So PDE can bind to lipidated cargoes that would go to the plasma membrane instead of the ciliary membrane, right? And there's an RL2 small GTPase that's regulating delivery of things into that membrane domain, likely because it also has unique activation at that membrane. So I think it's really about the activation of the small GTPase that's helping to sort of drive the delivery of the lipidated protein to specific sites. Yep. Yep. But, as you can see, we can do a lot with our system with electroporation, and we, as biochemists, we can do a lot in cells and in test tubes. But we really want to try to get to this question that Christoph just mentioned, which is that, like, what is actually the signal that's causing this migration defect? And so we started to think a little bit about what else could alter this gradient in rods. And is there something that we could use to get like a whole body knockout? Because this is basically, in order to do good biochemistry, we need to be able to sort of see this in the whole body. And we kind of came up with this hypothesis that the other way we could get to this would be by actually losing RP2, right? So RP2 is the gap for RL3. This is what's going to actually hydrolyze RL3 GTP back to GDP in an inactive form. And we all know that human mutations in the RP2 gene, RP2 stands for retinitis pigmentosa 2, cause X-linked retinitis pigmentosa. And so this seemed like a very obvious thing to check to see whether or not RP2 had a defect. So we acquired these mice from a non-spar group. We derived them. I think they got them from sperm. And we just confirmed that we have an RP2 knockout mouse here. This was made about 15 years ago, this mouse was originally. And then we wanted to see whether or not during these early development time points, we could see a defect in nuclear migration. And indeed, we can see a staining here. This is phosphorescent. This will label the whole photoreceptor cell body. We have a very nice displacement of rod nuclei now sitting in the internuclear layer with these little asterisks I'm showing here. And so this shows that these cells are in the wrong location. And this looked very similar to us as what was shown for the Q71 transgenic mouse, where you also just had this small population of photoreceptors sitting in the wrong layer. And so we thought, okay, well, when are these cells showing up? When are they getting there? And a new student in the lab, Dacia Flores, is starting to look at this pretty closely, characterizing when the nuclei start to actually show up in the internuclear layer. And she's doing first histological sections where she's counting the number of misplaced rods that are sitting in the internuclear layer here. And you can see in the RP2 null mice, we have quite an elevation of misplaced rods at even very early time points. This is sort of P7. At this time point, you can see that the peripheral retina is still sort of figuring out its boundaries at P7. It hasn't quite formed all of its layers yet. So you still have a lot of misplaced rods out in the periphery because development goes from center to periphery. But throughout the entire retina, you have very nice elevated misplacement of rod nuclei in the RP2 null mice. But, when you look at this, it looks like it's just a small population of rods that are mismigrated. So do all of these rods have a defective nuclear migration occurring in them? And so we decided to do some live imaging with our electroporation technique. So this is showing some stay gold. This is a fluorophore that's very photostable, so we can image it over the course of many, many hours. And so we did some electroporations. This is just from a wild type. We've just started getting some of this going. This is just showing you an on-face view a little bit, so you can see that we're kind of collecting a patch of these cells, and we're doing a Z-stack from a sort of full flat-mounted retina, and trying to get as deep as we can to see the nuclei as they're moving. And we do this recording for about 16 hours every 10 minutes. And so if you look at the side view here, you can start to see these nuclei actually moving. And we're kind of tracking them with these little blue balls that we put at the apical edge of the nuclei so that we can kind of move them up and down. And you're seeing some of these tracks coming off the back end of them. And we're tracking cells as they're moving. And this is just a wild type here. So you can see a nice ones that are moving over here and over here. And this is looping, but it's every 10 minutes. And so it's about an hour and 20 minutes a second or something like that, I believe, to get these movements like this. And we've just started doing some of this work, but so far from the sort of 41 nuclei we've tracked in this wild type animal, we have about 17% of the cells that we can actually see making these migration movements. And this is at a P6 time point. So some of these cells have already moved and are done moving, and other ones are still sort of like bouncing and getting up to where they belong. And just so you can kind of see this, because I know that this is very busy, there's a lot of cells here. I did put a couple just single ones here, so you can kind of just see the way that we're looking for these apical movements, right? We can kind of see beautifully the nuclei actually moving up like this. And we're very excited to use this system to begin to ask the questions of like what if we now knock down different signals that could, or different lipidated proteins within these contexts, how does that change these nuclear migration movements at this level? But also we can do this electroporations in the RP2 knockout mice. So we just got this data just the other day. We got a very nice big patch of nuclei here. This is also a P6 time point. You turn them onto the side and you can see that they sort of move, and we were able to track a lot more nuclei in this image. 187 we were able to track, and only 6% of them performed any sort of apical movement. So it does seem like there's a diminishment in the ability for cells to move. We need to do more. These are ends of one, but we're excited about how much data we're getting out of this. And we're thinking a lot about different speeds. We're thinking about how we can actually sort of move it to the next point of figuring out what the signal is itself. But also the RP2 mouse, right, so is great because we can sort of scale this up. We can also do some biochemistry. But we also are just interested in this phenotype because it's such a unique thing to have rods in the wrong location, right? In the wrong, sitting in the internuclear layer. And what's very interesting in this RP2 mouse is as we sort of go through time and look at these displaced nuclei, we actually see that they're sort of being removed over the course of time. So here's the number that we see at P14. You know, you have sort of a dozen or so sitting out there that are misplaced. And so this is just sort of like a spider graph across, right? And then by P21, they're already sort of being reduced. We don't see quite as many of them. And by three months, they're really only just a handful of them that we see in each section. And so that gave us a thought that these cells might actually be sort of selectively removed. And we immediately thought about microglia and did some imaging here, and can actually see microglia engulfing some of the cell nuclei in the RP2 null mice here. And this is a P14 time point, which seems to be sort of the right place where we know the nuclei are done moving, sort of set where they belong. You've started to make your outer segment and such. And now you have microglia coming in and eating up these displaced rods in the wrong layer. I think this also sort of is why most likely these handful of displaced rods were sort of missed the first time this mouse was characterized back in the 2020, I think it was like 2012 or so, was because, you know, when you start looking at 1 month, there's not that many of them there anymore, sitting in the wrong layer. And so people sort of started their analysis at 1 month and went the other direction, went sort of late to look at the degeneration due to, you know, phototransduction defects. And so, you know, lipidated protein is also defective. I think GRK1 is downregulated in the RP2 null mouse. And so people were looking at sort of those consequences, but hadn't looked at this early developmental time course and had sort of missed the nuclei displacement. And we also, of course, are interested in the idea of being able to actually restore this migration phenotype in RP2 as well. Like if this is really an overlap between these two genetic mutations, we would hope that we can actually be able to use some of the same tools we use in our RO3 data to restore the nuclear migration in RP2 mice. And this is a project being taken on by an undergrad in the lab. And so the idea is like just using our electroporation, just quick and dirty, can we track the number of misplaced RP2 misplaced somas in the RP2 null mice? And we can see quite nicely how robust this phenotype is. We can see quite a few of them misplaced, and this is just expressing an M-cherry fluorophore in the cells and looking at the placement of the nuclei using the same sort of tools that we had built for the RL3 project. And we have a very nice difference here between a wild type and our RP2 nulls. And so then we can go in and we can re-express some of these chaperones that we tried in the RL3 work and see whether or not that can also restore the nuclear migration in RP2. In the RP2 mouse model. So here is just sort of reminding you that we're dealing again with chaperones and having overly active RL3 sitting down on chaperones and preventing them from picking up lipidated cargos. And so you can imagine that by putting in more RL3 effectors, and we chose UNC119 and PDE delta, that we then also were able to restore this migration phenotype in these contexts. So we're very excited about the idea of really the fact that our work is showing that these two seemingly kind of disparate human diseases, right? Autosomal dominant RL3 and X-linked RP2 and it's being supposed to have a very similar underlying pathology where they have an overly active RL3 sitting in the photoreceptor. We can restore that using similar techniques, overexpression of GDI chaperones. We're now tracking these movements to try to get a better sense. I think that we're seeing already diminished number of cells that have this translocation ability. And that may be sort of stochastic just to the amount of RL3 that's expressed at a given time, and how important that signal is at a given time. And then we're also curious about what these misplaced nuclei are doing and if they have signaling capacity, why they're being selected for removal. Microglia are obviously going in and eating them up. We're interested in whether or not they can still form synapses, and we're looking at some of that closely. But I hope to be able to come back in a couple of years and tell you the exact signaling molecule that occurs from the baby cilia. But from my searching, and nobody's really looked at whether or not the photoreceptor cilia has any signaling capacity during these early developmental time points. And I think the sky's the limit. We're excited about it. And with that, this is the lab, and I will take other questions.
About cones.
Yes.
Because obviously they are just one layer.
Yes.
Is the same mechanism, or what is the other mechanism that hold them there?
Yeah, it does not appear to be about cones. I was ready for this question. So we've started looking at cones in the RP2 model. Obviously, our electroporation, you can't electroporate cones, so we were sort of limited in that capacity before. So we started looking at the RP2 null mice, and this is sort of the P10 to P14 time point is when cones are finishing their migration. You can see that they're still sort of like coming up here in the wild type. And we're using cone arrestin to just label. And what's nice is since there's so few cones in in the mouse, you can actually go through and just count them and use the same algorithm that we had generated for our electroporated patches. And so we can see that we have a kind of interesting phenotype where it's almost the opposite, where we have more apically displaced cone nuclei. And I think that this is also just a product of the fact that you have more basal placement of your rods, that now your cones are sort of non-autonomously being pushed up. I don't believe they have the same signaling, intrinsic signaling pathway being affected by RP2 and RL3 activity, because otherwise we would expect them to be basally displaced. So I think it's just a consequence of RP2 defect in the rods. Yeah. Yes.
Hey, Julian, really nice talk. Beautiful data. So my question is regarding the dominant allele. So the phenotype in the patient is still degeneration, I'm assuming. The photoreceptor degeneration, right? The RF3 dominant allele. But your talk really showed the also important for migration, but
right.
Only probably small portion will have migrate into the inner nuclear, get chewed up. So how you reconcile the phenotype of the primary degeneration phenotype versus this migration?
Yeah. Yep. I think that this is where we're also headed with the RP2. Now that we have a global knockout where we can look at all the cells across the whole retina, we can ask these questions a little bit better and ask the questions of how is misplaced rod nuclei affecting, the long-term health of the retina, as well as changing things in terms of if we restore the migration phenotype, do things still degenerate, right? That's a big question that we have. And we have plans to do. So now that we've identified that some of these chaperone proteins can restore the migration phenotype by electroporation, our plan is to do AAVs and restore it at that early time point, and then ask the question, how does that affect long-term degeneration, which does occur in RP2 mice? You have degeneration of the photoreceptors out longer, and does that help to restore some of that or not? It's a great question. We don't know. I mean, it's interesting because if you look at some of the original work with the mouse, the RP2 mouse, there are some sort of telltale signs that these population of cells is probably not functioning correctly because ERGs are quite low, even at that 1 month time point. You've lost about 10 or 15% of your ERG. So it seems like functionality is definitely diminished at those early time points. But I think to be determined as to how the two sort of interplay. Yeah. Yes.
Great work. I just have a few questions. These ARP mutation, do they affect just the primary cilia or motile cilia? And is the same interaction work in the motile cilia? And the patient, do they have other symptom phenotypes other than blindness?
Right. Okay, so the question is about RL3 mutations. Is that right? Yeah. Okay. And pathology in humans and syndromic possibilities, right? So all of these human mutations seem to be non-syndromic and having just RP, retinitis pigmentosa, which is quite interesting. That's also very consistent with the sort of RP2 patient population. So it doesn't seem like this pathway is important for any other cell types, nuclear mobility, that part for sure. And it's also very interesting to think that like maybe lipidated enrichment isn't that critical to cilia either. And I think the point here is that you can get a lot of the lipid protein just into the cilia on its own, right? just by sort of natural ability to sort of like get it onto membranes, and then it can get into the cilia. And so I think really RL3's role is about enriching that population, which some cell types are going to be a lot more dependent on for their function, which is photoreceptors, of course. They tend to have a big cilia and a lot of important components that must get there. So none of these patients have syndromic issues. So that's the best I can do. Is it? Mm-hmm. Yeah. 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 I mean, but so the BBsome, of course, and IFT and things, that's going to be much more in terms of getting the cilia even built, right? In order to actually build the cilia, you need to make sure that you have IFT components and stuff. So you're going to have a much more severe phenotype without a cilia. Whereas here, this is really sort of secondary, after the cilia is already built, you're just trying to get a population of lipidated proteins in there. So some of the cargoes that are reduced in primary cilia is actually INPP5E, NPHP3. Some of these lipidated proteins are reduced when you have a knockout of RL3 or you disrupt RL3 dominant mutations of RL3. So there absolutely is consequences, but it doesn't seem to play out in a horrible way for humans. No, it doesn't affect the formation. No, ciliogenesis is normal. That's right. Yeah. Yes.
Thank you for your wonderful presentation. I was wondering with the different variants of RL3 or the the new paper that came out, has anyone looked into like organoid models to investigate this?
Yeah. That's exactly. So this new paper that came out by Cheatham and Afridi's lab, they showed some very nice organoid model of their mutant, and I'm not sure I'm going to remember the mutation. It's G70 something of RL3, and they showed in that model in the organoids when they kind of grew sort of these rudimentary little outer segment that they had reduction in PDE delta. So that plays out exactly as you would expect for mature photoreceptors. They didn't really look at migration at all and nuclear migration at that point. So something that we'd like to test, and we plan to sort of throw that mutation in our system since we kind of have that going. Yeah. Yes.
So you're showing that these nuclei are not migrating apically as much as in the wild type, and also in the dynein knockout, you don't have as much apical displacement.
Yes, right.
If I remember my basic cell biology correctly, dynein is a one-way motor. So is there a signal that is actually actively pushing these nuclei basally?
Yeah, I should probably have been a little bit more clear about this. I apologize. But so in the paper that first looked at these nuclear migrations of rods, they really showed that the basal displacement, the downward movement of the rods was not motor dependent. It was just sort of a processive sort of like movement. And I think the idea there is that as cells are just being born, they're being born like at the top, and then things are just sort of getting pushed down as more things are being sort of born. And so there's like this tendency to kind of get pushed. And then once you're outside of sort of region of area, you need to get yanked back up into the nuclear layer is sort of the idea. I think the model is working at the moment, that you kind of get pushed down and then you have to get yanked back up by dynein. And so yeah, we are actively looking at how RL3 is and RL3 dependent ciliary cargo is regulating dining for sure. I think that's the angle we're looking at for sure. That makes the most sense.
Hi, yes, my question is about understanding how these mutations are affecting the interactions with effectors. So for the tyrosine mutation, tyrosine 90 from the structure that you showed, it seems to be in the interior part of the of the protein. So I'm wondering how it's impacting the interface with the GEF or with the effectors. Do we know what's going on like structurally?
Yep. That's a great question. So yeah, most of the dominant human mutations are found sort of on the core of the protein, oftentimes around where nucleotides are being bound, right? So that's sort of constitutively active is most of those mutations are sort of locking the GTP in, so you can't actually exchange for GDP, right? And so that was what a lot of our work was showing, was that the Y90C mutation is basically, affecting magnesium's ability to actually lock things in, and nucleotide is sort of escaping quite quickly. And so all of those don't have a lot of effects on the actual effector interfaces, right? Where RL3 would be binding to chaperones or its GAP or its GEF, right? Those are sort of exterior binding sites. However, they do ultimately affect the ability of RL3 to bind to different effectors based on its activity, right? And so that was like a lot of the assays were showing is that, like, you now have increased level of binding to the different effectors when you have too much activity, right? And likewise, like if you're just bound all the time to GTP with the Q71L or the D67V, you're always bound to GTP, then you're going to also bind to RP2 and just hold tight to RP2, because RP2 is not going to be able to hydrolyze. And RP2 binds to an active form of RL3, but it won't bind to an inactive form. And its job is to inactivate it. So typically this binding is very, very fast and transient, right? So it binds to RP2, and then it just sort of quickly becomes inactive and then is released. And so in those constitutively active versions, you then have it just bound and held on to the RP2 molecule, right? So absolutely, each one of these mutations is going to affect the effector population a little bit differently. And most of that stuff we just can do, by biochemistry in cells. But yeah.
Hi.
Yes.
My name is Rafał. I'm postdoc in Krzysztof Palczewski lab. I have a question. Do the patients or the animals have a hearing phenotype when the ARL2 or RP2 are mutated?
They do not. No hearing phenotype. Nope. Yeah, that's an often crossover between some of these ciliary proteins, but not this one. So, yep.
Hey, Jillian, beautiful work. I have two questions.
Okay.
We showed with my colleague Didier Hodzik a few years ago that disrupting the nuclear envelope, the link complex, Nesprin and lamin B1, B2, disrupts the nuclear migration of cone photoreceptors, and you end up with a retina where the cones are now at the basal part of the outer nuclear layer. So have you looked at possible interactions between RL3 or RP2 and nuclear envelope proteins? Yeah,
that's a really beautiful work. Yes, I remember this very well, and the co-nuclear migration work is strong, and I like this. We have not gone looking at some of those things yet. We're really taking an unbiased approach initially, just to see what's changed in the RP2 in the proteomes, both just of the whole cell. So we're collecting whole rods, and we're also collecting proteomics of just the cilia as well, to see if there is a signal specifically that's altered. So I'm hoping that we can get there in an unbiased way, but if not, we will absolutely go after some of those targets, and test them as well. Yeah.
Okay, thank you. That makes sense. And my second question is, I guess in the RP2, you showed that there is reduced migration of the rod nuclei, but they still end up in the outer nuclear layer.
Most of them still do.
Any idea why they degenerate? And specifically, have you thought of restoring RP2 expression in the adult to see whether if you restore the delivery of lipidated proteins, you will prevent the degeneration?
Right. Yeah, no, these are great questions. So the first one, right? Degeneration, right? So yeah, it does seem like that was something that would keep me up at night, is the fact that most of these rods are just fine, right? They're fine. They're in their outer nuclear layer. And if we look, they polarize fine. They have outer segments. Like everything looks really, really good with them, So it seems to me like, because rods are sort of the last ones born and they don't have to really go that far. They probably don't have the same sort of migration where they're going all the way down to the ganglion cells. We're not doing those sort of crazy things. And we're not really seeing that in our live imaging either. We're seeing sort of some displacement, and then they're kind of coming back up, and a little bit of displacement and coming back up. So that helped me sort of process this a little bit better, in the fact that like most of them are probably okay. And so some of the work has actually done some gene therapy in RP2 mice, right, where they've restored RP2 back in adults, in both rods and then in cones specifically, because there's a cone phenotype in that mouse, which we're going to look at too. But and they do have rescue to some level, but never back to wild type, right? Never back to like perfect levels. And I think that is really because they've lost a 10 or 15% of their rods right out the gate, right? They just don't have as many photoreceptors anymore. So they never get their ERGs back to a wild type level, even when you put RP2 back in at 3 months or something like that.
So then, as a follow-up, do you think that the degeneration is caused by nuclear migration disruption, or is it really the delivery of lipidated proteins that is the essential problem?
Right. I think probably ultimately long-term degeneration is caused by lipidated transport into the outer segment, for sure. I think that would be like the mature photoreceptor phenotype. But I do think that there is a component in these early developmental time points that is now sort of being revealed to be important. And if you actually look at the patient cohort, RP2s are some of the youngest patients coming in with issues. So really, I think that's sort of is what we're getting at, is like there's kind of two waves, right? You have this first wave where things are not in the right place, they gobble up those wrong misplaced nuclei, those misplaced rods, who are sort of then at a little bit of a deficit, and then moving forward, you degenerate. Yeah.
All right. Anybody brave on Zoom?
I have a
It
chat.
looks like we don't. So thank you so much.
Yep. Thank you.